Hello, everyone. So I'm sure that you're all familiar with organelles. You know, those small bodies in cells, such as mitochondria and chloroplasts, that perform essential cell processes such as photosynthesis and respiration. I'm sure that you're also aware that these organelles used to be free-living bacteria that became imprisoned within cells. What you might not know, though, is how exactly these imprisoned bacteria transitioned to organelles. What happened to their genomes after they were engulfed by cells? How were their genes lost? What are the functions of the genes that were lost? This talk sheds some light on these questions by summarizing the findings of a recent research paper in this area. So where did cell organelles come from? It is theorized that long ago, a proto-eukaryotic cell that consumes bacteria engulfed anaerobic bacterium, and over time, this bacterium became the mitochondrion. Later down the line, some of these cells then engulfed photosynthetic prokaryotes, resulting in the birth of the chloroplast. So to come back to the main point of this talk, once these bacteria were internalized, how did they then transition to lifeless organelles? Well, a recent research paper has attempted to answer this question. The study focuses on the obligate endosymbiont of Epithemia turgida, which is a diatom. A diatom is a microalgae found in various wet environments such as oceans and soils. An endosymbiont is an organism that lives inside another cell and has a mutualistic relationship with the host cell. This diatom endosymbiont, which is called a spheroid body, recently adapted to an intracellular lifestyle, and so may be in transition to becoming an organelle. In the paper, these adaptations were studied by analyzing the spheroid body's complete genome and comparing it to other free-living species. Firstly, the researchers compared the genome size and composition of the spheroid body to that of two related cyanobacteria that are free-living. As you can see, the total size of the genome is massively reduced in the spheroid body. The number of protein encoding genes has also gone down, which suggests that many genes are no longer required in organisms that evolve intracellular lifestyles. What's most interesting, though, is that the number of pseudogenes has gone up in the endosymbiont. Pseudogenes are remnants of active genes that became inactive as the organism evolved. The fact that the pseudogene count has gone up in the endosymbiont suggests that gene reduction is in fact ongoing. But what are the functions of the genes that have been lost in the endosymbiont? This graph on the right shows genes that are involved in many essential cell processes, such as signal transduction and metabolism, have been severely reduced. The endosymbiont is thus clearly becoming more dependent on the diatom and is clearly transitioning to an organelle. Okay, so we know that genes are being reduced in the endosymbiont. Organelles that exist today have evolved to become specialized in specific functions and lack almost all genes found in free-living bacteria. What kind of organelle could this spheroid body be becoming? We know that it is related to cyanobacteria, so a chloroplast may seem like the obvious answer. The problem is that the spheroid body is non-photosynthetic according to the research. As you can see, there are photosynthetic genes and complexes that have become inactivated, such as the genes for the enzyme rubisco, for the enzyme rubisco, which performs the essential carbon fixation step of the Calvin cycle in photosynthesis. Membrane complexes involved in electron transport in photosynthesis have also been inactivated, according to the research. Hence, this spheroid body is likely not becoming a chloroplast, despite it being related to cyanobacteria. So what could it be becoming then? Well, according to the research paper, it appears to be becoming a nitrogen-fixing organelle. Nitrogen fixation involves the reduction of gaseous nitrogen to ammonia to produce biomass. The experiment to the right showed that when incubated with gaseous nitrogen labeled with 15N, which is a heavy nitrogen isotope, the Epithemia turgida protein chlorophyll A became much more enriched in 15N compared to that of diatoms that lack spheroid bodies. Hence, nitrogen must be being fixed by the endosymbiont. In addition, a complete set of genes that are known to be involved in nitrogen fixation was found in the spheroid body. And in contrast, many genes involved in respiration and photosynthesis are inactive. So overall, what can we learn from this paper about how internalized bacteria became organelles? Well, it appears that gene reduction occurred continuously until the bacterium lost all essential functions. bacterium also became specialized in a specific function, such as respiration, that is a benefit to the host cell. And that is the end of my talk. I hope you found this engaging and are now interested to learn more in this subject area in the future.